going to focus on the extracellular matrix component of this microenvironment. Interestingly, this same environment that supports hematopoietic stem cells to survive and provide hematopoiesis are the same mechanisms used by leukemia cells to protect what we call the leukemia stem cells. And leukemia stem cells hijack the bone marrow environment to their advantage and for their survival, and they use the same mechanisms that allow the hematopoietic stem cells to survive to their advantage. So, in studying the extracellular matrix, the tissue of interest, obviously, for this cohort of people has to be related to umbilical cord tissue. And it is related to it. It's Wharton's jelly matrix. And Wharton's jelly is this mucocutaneous tissue that keeps blood vessels patent in the cord tissue to allow blood supply between the mother and the baby. And without that tissue being providing that function, blood supply to the baby will be interrupted. And this tissue, when I started reading about it, I was actually isolating MSCs for the goal of creating 3D environment to support hematopoiesis. But by looking into this matrix, I got interested in it to the point that I started reading more about it and realized beside the MSC component, it has a matrix component. And a lot of these, if you look at them, collagen, hyaluronic acid, sulfated glucose aminoglycans, these actually are present in the bone and bone marrow environment. In addition, it has soluble factors, including peptide growth factors like insulin-like growth factor and platelet-derived growth factors. Why hyaluronic acid component is important? Because this is the receptor for CD44, which is present on hematopoietic cells. So this matrix actually can allow hematopoietic cells to adhere to it. And in addition, these factors that are present in this matrix can influence cell behavior. So now, in tissue engineering, if you start with a natural tissue, to create a matrix out of it, you have to go through the process of decellularization. So I applied that to our matrix, which is Wharton's jelly, and got decellularized Wharton's jelly matrix. And this was actually in collaboration with two scientists, one physician and one scientist, Dr. Hopkins and Dr. Dedamore. They really helped me get this accomplished. And the product of this work is what you see on the panel A, is decellularized matrix. It looks like shrimps, if you can think of it. And C, this is scanning microscopy, shows that this matrix has many lacunae or open spaces, so cells can go in. And in B, you can see it has a lot of, so it's porous. And when we did mass spectrometry of this matrix to study it, we did see that a lot of the components are present, still present in this decellularized tissue in terms of collagen, fibronectin, tenacin, lumbican, and glycose aminoglycans. And again, remember, these are the components that you see in bone and bone marrow microenvironment. So through work by Sushma Jadala-Nagari, who's now graduated, we looked at seeding this matrix again with the same cells that used to live there. And as you can see here, these cells were stained, and you can see them. Within 2 hours, they adhere to the matrix. Within 1 day, they start their processes, and within 2 days, they have the stromal cells go back to their normal state. So they like the matrix, their initial home. And I actually like this video to show, how can I show the video? I want to click, oh, here you go. Thank you. So as you can see here, in this video, what we try to do is, overnight, we added the cells to the matrix and we labeled both. And as you can see, the cells go in and out of this matrix. And one of the questions was, does the matrix move by doing that? And actually, as you can see, it did actually move. So to summarize this part of the presentation, We can apply decellularization processes to Wharton's jelly matrix, and we can get a matrix that has similar components to bone and bone marrow environment. And this matrix can allow stromal cells to interact and attach, and also allows for the invasion inside the matrix. So now, knowing that, we started to hypothesize, can we use this matrix to support hematopoietic stem cells, as well as leukemia stem cells, with the goal of, if it can support hematopoietic stem cells, it can expand them. If it can support leukemia stem cells, we can start understanding how these leukemia stem cells function. So obviously, as a physician, I always think of the end product at the end. So if we expand these cells, obviously we can use them for transplant. So this is what I do. And depending on the product that we expand, we could use it for different things. For leukemia stem cells, I'm thinking about it as if we use this model to culture leukemia stem cells, we can actually look for therapy that can overcome resistance in these leukemia cells. So that's how I think about it for the future. So let's go over these two applications for this matrix. So obviously this group knows that cord blood transplantation has very important advantages, but there are two drawbacks, and one of them is there's limited number sometimes in these cord blood units. But also biologically, there might be defects in how they home to the bone marrow, and that results in delayed engraftment, especially when it comes to megakaryocytes or platelets. As you know, when stem cells home to the bone marrow, they are driven by a chemokine inside the bone marrow, which is SDF1. And the stem cells have a receptor on the surface called CXCR4, which is really important for homing of these stem cells. So we need expression of CXCR4 to be able to have adequate and efficient homing. In the field, there are many efforts to try to address these two questions, and some of it has to do with processing of cord blood units at the time of collection, or expanding them, or improving homing. And I want to introduce this method now, which is using matrix as a way to expand hematopoietic stem cells. So again, the hypothesis is that we can use our matrix to expand hematopoietic stem cells. So this work was done by now Dan Dunley, who's graduated from my lab. And to simplify the model, I use this cartoon. And you can see here there are many components in this model. So we have the decellularized matrix, which is depicted in the blue fibers. And then cells either adhere to it directly, or we use in other controls, bone marrow stromal cells to support these stem cells. And when we analyze these cells, we look at them as the ones that remain in suspension or the ones that are adherent to the matrix. So this is another way to show the components. And you can see in C, we use sleeves of this matrix. We call them wafers at the time, thin fibers, thin cuts of the matrix. And in D, you can see the cells that remain in suspension versus the ones that adhere inside the matrix. And to be able to analyze those, we have to subject the matrix to a step which is using collagenase to get the cells out to analyze them. So one of the first things that we looked at is the ability to form colonies, and looked at all these different culture conditions. And as you can see, in the presence of matrix, we have the most number of colony-forming units, including erythroid as well as myeloid colonies. And actually adding the mesenchymal stromal cells to this culture condition reduced the amount of erythroid colonies. Now, we talked about how CXCR4 is important for homing of stem cells. And what we did here is we looked at expression of CXCR4 and CD34 in cells cultured in our matrix. And we did see in panel B that the highest expression of these two markers was in cells that were cultured in the matrix, especially the ones that remained in suspension compared to the ones that were adherent to the matrix. In panel C, we did something called transmigration assay. So what it is you look at the fraction of cells that migrate from the upper well to a lower well through fenestrated membrane with SDF1 in the lower part. And you can see the cells that migrated the most were the ones actually cultured in the matrix. And again, one of the problems in cord blood transplantation is time to platelet recovery is delayed. So what we looked at one of the platelet differentiation markers, which is CD41, and we did see that cells that were cultured in our matrix had higher expression of CD41. And when we did RNA-seq studies, we did show that there is increased expression of genes that are important for platelet differentiation. So if you think about it, by culturing the cells in our matrix, we can address the limitation in terms of cell dose, we can address homing, as well as we can address platelet engraftment. So to summarize this part, I would say first, this matrix seems to support hematopoietic stem cells in this case, cord blood CD34 cells. It does enhance their transmigration potential, clonogenic potential, as well as megakaryocytic differentiation potential. So hopefully we can use this in the future for transplantation to improve upon our results. Okay, now this is the first application. So let's shift gears to the second application, which has to do more with the leukemia stem cells. So for people who deal with leukemia as physicians, know that this is an important problem because in many times when we treat patients first with chemotherapy, we can get disease to a low level that the immune system can get rid of it, and these patients are typically cured. But unfortunately, a lot of them, we can't get to that level because of persistence of leukemia stem cells, and eventually these patients end up relapsing. And this is very common, unfortunately, in leukemia. And again, we think the reason why it happens is because when we treat patients with chemotherapy, we get rid of the bulk of the cells, but leukemia stem cells still survive. And these are supported by the microenvironment in the bone marrow. So to study these cells, you have to create the microenvironment to create them to start with. So our hypothesis from this work is if we grow these leukemia cells in this matrix, can we support leukemia stem cells? And again, Dandon Lee was the PhD student at the time who really did this work. So one of the characteristics of leukemia stem cells is that they are dormant. And what we've seen when we cultured cell lines in this matrix, that their proliferation actually goes down over time. And it's not because of viability issue, because the viability was still maintained in the matrix. One of the other features, or one of the other findings of our work is that when these cell lines were cultured in the matrix, these cells started to change in shape. And as you know, most of these cells are round, but in the matrix, they become actually elongated and stromal-like and spindle-shaped-like. And actually, most of them in our research were actually spindle-shaped. One of the markers that used to determine stemness is expression of ALDH. And when we did this in leukemia cell lines cultured in this matrix, we did see there was increase in the expression of ALDH. Also, one of the characteristics of leukemia stem and progenitor cells is that their ability to self-renew. So what we did is we did colony-forming assay, and we did see that cells cultured in the matrix, they actually form more colonies. Not only that, when we replated them again, they kept forming more colonies, which is indicative of their ability to self-renew. And finally, when we assessed their response to chemotherapy, we did see that these leukemia cells cultured in our matrix, they resisted the effects of chemotherapy and remained more viable compared to control conditions. So in summary of this, when we cultured leukemia cells in our matrix, we did see that these leukemia cells changed in their morphology. They had decreased proliferation and increased expression of ALDH increased colony-forming units, and they had more resistance to chemotherapy, which, when you think about it, these are characteristics of leukemia stem cells. Now, again, why this could be important, because understanding how leukemia stem cells function and the interaction with their niche can eventually help us in designing drugs that we can use to target these leukemia stem cells. So a few years ago, I moved from University of Kansas to University of Rochester and collaborated with a biomedical engineer. His name is Hani Awad. And what we did is we modified this matrix a little further. So initially, we started with this piece that looks like shrimps. We made wafers out of it. But what we did finally is we were able to create a form of it that is dry. And as you can see on the right side, we can actually make sheets out of that, and we can use punch biopsy kits to make actually discs out of it that we can use in our culture conditions. So what I will do next is show you some of the interesting things that we did with this new matrix. So one is we looked at how these cells adhere to the matrix, and for that used scanning electron microscopy. And these are actually patient leukemia samples, and what we did is we cultured these cells in this matrix for 3 days, and as you can see, these cells like the matrix, they adhere to it, they start to form small colonies. They look like grapes somewhere, and so they like the matrix. And we used confocal microscopy because this form of the matrix is so thin that you can actually use confocal microscopy to study things. And what I want to show you in this video is interaction between leukemia cell and stromal cells. The stromal cell in pink, leukemia cell is in green. And what you see is they both are adherent to a fiber of the matrix. If I can run the video, please. Oh, no. The previous one? Yeah, there is a video. Here we go. So one thing you can see for sure is that, number one, is the matrix itself actually moves. And the other thing is the cells, they seem to talk to each other. The stromal cells seem to be a little ballooned here, but it kind of allows you to see the scan interaction. Now, obviously, one of the things that we want to do using this matrix is to recapitulate the bone marrow environment. And in this confocal video, I show you leukemia cells and stromal cells labeled together. And as you can see in this, actually, there is a video. These cells keep moving, but there's a cluster of these leukemia cells and stromal cells, which is exactly what you would see in a bone marrow microenvironment. Now, this is one of the very interesting findings as of late in our model. So, since we started to study bone marrow microenvironment, we noticed what we call them naked nuclei. And so what we do when we do a bone marrow biopsy, we take a sample from the bone marrow and we put it on a slide and then smear it. And our suspicion was always that the smearing process is what creates these naked nuclei, so nuclei outside of the cells. And for the longest time, this was our understanding of why these things happen. It happens in normal bone marrow biopsies, but also happens in leukemias. It's more common in leukemias. Interestingly, it's also seen in solid cancers. So you can actually see that also in cases where you don't have to do the smearing part. So when we did our confocal studies, what we did is we stained cells with 3 stains. One is Host for the nucleus, and one is cell tracker green for the cytoplasm, and the third one is Sylvia maroon for the membrane. And after culturing the cells in the matrix, we digested the matrix and we looked at what's going on. And what we did notice is that, as you can see on the left side in the first 2 panels, you can actually see what we call them naked nuclei. So these are only positive for Host. They don't have a cytoplasm. They don't have a membrane. And then in the lower part, you can see different combinations. So you can see what look like cells that have the cytoplasm, but don't have a membrane stain. And then at the end, you can see cells that stain positive for everything. And on the right panel, what you can see is a timed video of interaction between these naked nuclei with cells that has nuclei and membrane. And over time, you can see them getting close to each other. And on the lower part, we just show over time how the distance between these two, the naked nuclei and the cells, they become close to each other and interact. So this is a new finding, and we have more follow-up studies to tell us more about the function of these naked nuclei. So in summary of this part, so matrix can be used ex vivo to expand cord blood CD34 cells. And again, these cells have improved transmigration. They have improved megakaryocytic differentiation. And also, we can use this matrix to induce leukemia stem cells that has drug-resistant phenotype. And also, like I showed you in the last few slides, we can use this model to study more of the functions of leukemia stem cells, and hopefully we can have more discoveries, like I showed you earlier about the naked nuclei and how we can actually induce that through culture in this matrix. I want to thank many people who really helped with this work. This is a work that probably occurred in the last 7 to 10 years from the start to now. And there's a long list of people who really helped make this work and make it happen. I think that brings me to the last slide. Thank you. So what we'll do is we'll go for quick questions, and then at the end we can also have a discussion. Megan.
So did you at all look at whether or not there was a change in the membrane polarity? Like, by being in culture, they lose the polarity, but by being bound in a 3D like matrix, it maintains that polarity, thus maintaining their function. And on like a connected note, do you see at all a change in the amount of, in your transwell assays, and the amount of SDF? So like a lowering of the threshold of chemokine necessary for that recruitment because of changes in the membrane?
These are all great questions. So to start with, polarity is something I've seen for sure. And if I can go back to my slides, is there a way? I want to show you one thing. Thank you. But I agree there is polarity that we see. I'm going to show you one example of that. If you do the last one, and then I'll go back. Thank you. Yep. Okay, yep, sorry. So if you look at so the left side panel, if you look at third part, you can see a little polarity there. So the nucleus seems to be like to one side and the cytoplasm is to the other side. And I have a feeling this is an event that happens before the nucleus actually exits. I did show earlier how the MSCs react with the leukemia cells, and they look polar. So I think there is polarity that happens for sure. Now, in terms of the other question about SDF1, we haven't tried lower concentrations of SDF1.
Yeah, you should maybe throw on some cholera toxin on there to look at the lipid rafts to see if that's in that little like polarized pocket, and so therefore you're changing the threshold of growth factor and cytokines needed to maintain. Yeah.
That's a great suggestion. We can talk about that for sure.
Just two really quick questions, if it's okay. So you showed the increased transmigratory potential. Have you actually tested it like in a mouse model?
Have not yet done that.
If you want to collaborate, we'd
yes.
love to.
Yes.
It's a really cool model.
I'm happy to do that for sure.
And just a quick follow-up. So it's a really cool way to model the extracellular matrix. Have you thought about adding in other things that would be different in the bone marrow niche, potentially lower oxygen tensions, some of the other factors that might be present there, those types of things?
For sure, yeah. I want to say this is just the start. And I have another model that we started with where we had synthetic matrix and stromal cells, and we actually studied that before this one. But then I thought it's easier to start from scratch and just have one cell, one matrix. But like you said, this is the next iteration, adding other things. I agree with that for sure. Yeah.
Thanks.
Thank you. That's a great suggestion. Okay, so we can move on to the second talk, and this is by Dr. Krish Roy from Georgia Tech. He's going to present about preclinical development of MSC therapies.
Thank you, Omar. I wish I knew what you're speaking today. I could have brought some bone marrow slides. We have published on a bone marrow model recently, last year. But today what I'm going to talk about really is really scaling up from an engineering standpoint about manufacturing either cord tissue-derived cells or bone marrow-derived cells. I think I last spoke to this meeting about 3 years back. Since then, there's been quite a few developments, and I want to talk a little bit about that. Okay, so I think to this audience, cell therapy is really exponentially increasing, but we have three fundamental problems. That is of manufacturing capacity and scale, cost, and then quality. I think you all have seen that even for FDA-approved products like the myeloma CAR T therapy, patients are dying in line, even though there is an approved therapy available. Neither of the companies can make enough. So there are patients, maybe 3 or 4, that are treated in a single clinical center at a time. Obviously, the costs are tremendous. And then the last one, I was really hoping we'll hear from Mesoblast today. But even when advisory panels approves a therapy for 9-to-1 vote, the FDA rejects it. And we will hear many things from Peter Marks tomorrow morning. I hope he addresses some of the quality questions as we go along, because quality, reproducibility, and our ability to predict quality is going to be a paramount feature that the FDA is going to look for when we get into this approval phases of our therapies. So we have been really fortunate to receive funding from the National Science Foundation to develop an engineering research center that's across 8 US universities, 4 international universities, and as I would show you, many different companies. And parallel to that, at least at Georgia Tech, we have been able to receive philanthropic funding similar to what Joanne has received at Duke from the Marcus Foundation to really create a large ecosystem of clinicians, biologists, engineers, scientists, physicists, chemists, working together to solve the problem of scalable manufacturing and reproducible quality in cell therapy. So these are the folks who are part of that center. On the top are really the scientific and engineering expertise, and on the bottom are our clinical partners. We developed a formal private-public partnership with the FDA, with CBER, with NIST. We work very closely in developing the early standards for cell therapy manufacturing, anything from cell counting to flow cytometry to rapid microbial detection. There are many different standards that are now going through the ISO and ASTM process. And if anyone wants to participate in that, please reach out. There's a tremendous need for industry standards in this space. And obviously, we work very closely with patient advocates and reimbursement experts as well. These are our industry partners. I think the CME folks was sort of, is this allowed? But then they allowed it because it's many different companies together. But these are our industry partners, from large pharma and small biotechs to startups. Everyone really laser focused on technologies for scalable manufacturing of cell therapies from various sources. So what is quality by design? And this is really a regulatory term that has been used for decades now by the FDA. It's a systematic approach to biopharma manufacturing. And the key premise of that you know your process so very well that you know what is the impact of every component, every reagent, every petri dish, every tweak, every knob, and how does that impact the quality of the cell and the performance of the cell when we inject it into the patient. And the FDA uses 3 terms: critical quality attributes, critical process parameters, and mechanisms of action. So mechanisms of actions will get there by the time one gets to phase 3 or BLA applications. You need to show potency assays. You need to understand mechanisms of actions. But very early on, even in process development, one needs to address what are the most critical attributes that I need to reproducibly make every time I manufacture the cell, wherever I manufacture it, right? So are those a certain amount of genes, certain amount of protein, certain lipids, certain metabolites? What is that combination of things? And what are the process parameters that control that? So from our point of view, cell therapy and manufacturing of cells at a scalable manner is incredibly complex. We, as a human civilization, has never done that. We have manufactured plenty of microchips and cars and planes and boats and everything else, but never really a living material at scale to treat hundreds and thousands of patients, right? And the bigger problem of this is that the starting material is incredibly variable. All the cells that we get from any one of us would be very different from cells that we start with from someone else. And it varies across all these parameters. And we take that and we put what is currently a fixed manufacturing process. We guard our manufacturing process to the T, and nothing can change into that, and the FDA requires almost that. But if you take a variable material, put it in a fixed manufacturing process, biology and physics dictates that we will get a variable product every time we manufacture it. There is no if, don'ts, and buts about it. And the other question that prompts, especially from a regulatory standpoint, is that what among that goop that we are injecting, the millions and billions of cells we are injecting that are very heterogeneous, what is that most bioactive molecule or the set of bioactive molecule that is critical for cerebral palsy or autism or cancer or arthritis or whatever that is? Is it a combination of RNAs? Is it a combination of RNA and metabolites or protein? There are probably 1,000 different drugs, quote unquote drugs, that we are injecting on the patient at the same time, and we have no clue what the PK/PD of any one of them are. And the bigger issue is that pharmacokinetic pharmacodynamics and the relative amount of the RNAs and DNAs and metabolites and lipids changes with the status of the patient. What chemotherapy did they have? What disease condition do they have? What is their immune status? All of that influences the cells inside the body. So in this really critical dynamic situation, how do we say what is the best potency assay? What is the most predictable feature of the cells that will tell us that we will get an 80% or a 90% success rate in a particular patient's strata? We don't know any of that. And that is, eventually is going to catch up on us when the effect sizes are not going to be as large as CAR T cells, right? So we need to think about that. So how do we define quality of a particular cell therapy or a cell product? What are their critical quality attributes that we need to reproducibly manufacture? What are the critical process parameters that controls those quality attributes? Those are all unanswered questions in the cell therapy space. What is the most important subset? In the CAR T space, it is increasingly now understood that only very, very few clones of the billion cells that's being injected actually stays and is actually the most functional part. So probably 900 million of the cells that we are injecting didn't need it to be injected. But we don't really know what that little subset is very well right now. And that is a very critical scientific process that we need to think about. So I'll talk about 3 vignettes coming from an engineering and scalability manufacturing standpoint. One is that of quality really understanding and discovering critical quality attributes, which is not an easy task when you have a very complex product like cells. So this is kind of the method we use. As I showed earlier, we have cells that goes into a manufacturing process that creates a product. Sometimes this manufacturing process could be 4 weeks long, 8 weeks long, even 12 weeks long for iPSC-derived cells, right? And then that manufactured product goes into a patient. So we use really a broad array of multi-omics characterization of the cells, of the product, the starting cell, the product, as well as the patient, and then use really AI-type modeling approaches and correlative modeling approaches to say these are the parameters that best correlate with product quality, and these are the product quality that best correlate with patient outcome. And I quote here Patrick Hanley, who I know he's here, quoting Peter Marks a few years ago at the ISCT meeting, and Peter is essentially saying that in an ideal world, we'll have supercomputers crunching all of these omics data, characterization data, patient data, and really spewing out what are the most correlative factors. That's exactly what we have been doing for the past few years and continue to do in many of the critical cell therapy applications. So this is our process development workflow. We do a lot of design of experiment study to look at multivariate spaces. We do multi-omics analysis from lipidomics, metabolomics, proteomics, transcriptomics, epigenome, and then understand product function, product assays, then come back and reiterate the process parameters to get to a certain product quality. And we are very agnostic to the mathematical approach that we take, from linear modeling to nonlinear modeling to AI algorithms and so on. So I don't have time to really go through this. This is an example for T cells, but we have now applied that for MSC products as well, and really looking at a variety of process parameters. Oh, this is interesting. I didn't realize that. So we look at variety of process parameters, then looking at those process parameters and the functional outputs of the product at the end of the manufacturing, and simply ask this question: how early in the manufacturing process, by measuring the culture, can I tell what my product will look like at day 14? 4 weeks later, 6 weeks later. One of the big things that industry wants us to tell them is, how early can I tell that I have a bad product coming up? Because I don't want to take up a GMP room for 4 weeks and 6 weeks, and at the end of that process find out that I have a batch failure, because that's $5 million just down the drain, and that cost gets passed down to the consumer. But more importantly, if batch failure in cell therapy often means a dead patient. So I think this is a critical question that we have been trying to answer. And what we generally do is really look at every process parameter, how varying that process parameter changes product quality, from cell numbers to cell phenotypes to markers, marker ratios, and then find consensus across various modeling techniques that tells us, okay, IL-2 receptor or TNF-alpha at day 4 of the culture is predictive how many memory T cells we are going to get at the end of day 14 manufacturing. And we use this to create models and then validate those models. So in process, in real time, during manufacturing, we understand that the culture is going in the right direction. And we have done this now both for a T cell product that we recently published, for an MSC product in this case, looking at metabolomics and secretomics, to talk about how MSCs suppress T cell function, and can we predict that very, very early on. And then very recently, looking at the headspace gas chromatography during MSC culture to show immunosuppressive properties of these MSCs can be predicted by looking at simply the volatile metabolites early on during culture. And so these are now allowing us to monitor quality and, if necessary, take action during the culture period to get to a certain product quality at the end of the culture. So I will not go through this. This is published work, so you can look at it. But the basic of this is that headspace gas chromatography is a non-destructive tool. You can have a probe sitting on the top of your culture, and it's simply measuring what's volatile metabolites out of that, not even dipping into the culture space or the media. And that goes into a GC-GC mass spec, and you look at the signature of that, and you look at volatile metabolites. And measuring that, we showed that we can monitor cell proliferation. On the bottom left, you will see and this was very unexpected, somewhat serendipitous. We found that benzaldehyde decreases as MSCs proliferate. And in batches where we had an unproliferative MSC, the benzaldehyde was not really decreasing at the same rate that it will decrease for cord tissue or bone marrow, where we have successful expansion. We don't know what. We have tried to look out metabolic pathways yet, but we are looking, where that pathway might be. But we have also looked at, and I only show the T cell suppression data by MSCs, but we have looked at for IDO production and other functions, where you can actually find very correlative volatile metabolites that correlate with high T cell suppression function of MSC. So now, during culture, without even doing a suppression assay, one can start monitoring the quality of those cells over time and get to a certain specific product quality as we try to scale up production. So this has led to something that many other groups, including Joanne's group, participate in. This is an osteoarthritis clinical trial across 5 hospitals, comparing adipose-stromal vascular fraction, cord tissue MSCs, and bone marrow aspirate concentrate. Emory is the lead of that. 480-patient trial, 120 patients in each arm, a steroid arm as a control. This unveils very soon, in about a month. We would know if any of these has any effect on osteoarthritis or has 0 effect on osteoarthritis. At least we will know that. But then what we are doing as sort of the engineering characterization node is all of these cell samples and patient synovial fluid samples are shipped to Georgia Tech. We do all of these assays from transcriptome, metabolome, lipidome, functional assays on them. And as the clinical trial data gets unveiled, we are poised to do correlative modeling to see what substrata of patient might benefit, whether there is an age, sex, ethnicity variance in there, whether there is a KL score variance in there, or what data of the cellular properties most correlative to patient outcome. This led to funding from CBER. FDA decided to fund a subset of this study. They asked us to take 50 patients worth of data out of it, and then do the correlative modeling based on AI type algorithms to correlate patient, cell properties to patient outcome. But at the same time, they also asked to do 2 other things. Can you find an in vitro potency assay that is predictive of the clinical outcome, number 1. And number 2, can you find a preclinical animal model that is predictive of the clinical outcome? And so those are very high bars, right? But this is something regulators are looking for. Is your potency assay anything to do with the clinical outcome? And if you look at the FDA document from the Mesoblast filing, the BLA filing, that's what they were asking, that you had your potency assay has no correlation to your clinical outcome. So this is not a valid potency assay anymore. Now, we can all argue about that to the end, but at the end of the day, that's a bar we have been dealt with, right? So these are still very, very unanswered questions, and we are working on them. Hopefully, in about a year, we will have these data fully analyzed. This has also led to NIH wanting to do the same thing. We are now at Georgia Tech, the first in-depth cell characterization hub of the National Institutes of Health. This is part of their Regenerative Medicine Innovation Program. What this means that anyone who is funded through the RMIP program to do trial, preclinical and clinical trial, must send their cell samples to Georgia Tech for detailed multi-omics analysis, and also submit their clinical trial data for correlative modeling work. We are just starting that. There are 12 RMIP projects funded by NIH, anything from iPSCs to bone marrow aspirates to fibroblasts, and we are working with all of those investigators around the country to provide that as a service to NIH. Finally, in the last sort of 5 minutes, I want to talk a little bit about kind of the scalability. As we go from 10,000 patient to 20,000 patient to 100,000 patient to 1 million patient, how are we going to make these cells, and how are we going to automate these cell processes? So Ideally, as I said, this is our current bus. It's a non-responsive fixed process. We have a variable input. We get batch 1, batch 2, batch 3. Each of them are slightly to a lot different. We very rarely characterize even that. It goes into a patient who is also a variable, and then we get a heterogeneous response in all clinical trials. The goal here is to now have a flexible process where we monitor quality in real time and then feedback and modulate the process to get to a much more uniform consistency from batch to batch, so we finally get to a more homogeneous and hopefully a better clinical response. So we have been doing a lot of chemical modeling work. This is sort of the domain of chemical engineers. This is bioreactor in a hollow fiber, starting with MSC, cord tissue MSCs, for example. And then we have modeled sort of the glucose lactate level to avoid the fluctuations of glucose lactate levels and really keep a steady glucose level. So what we do is that we have multiple sensors around the bioreactor. It monitors in real time, and then we create a mathematical model that models the glucose consumption rate, the measurements rate, the infusion rate, and we continuously infuse media and glucose or lactate, or for that matter, any other cytokines or growth factors, and try to keep a very steady bioreactor conditions over time. So this is a feedback control. We sense, we decide, we control automation platform. So this is how it looks like. This is kind of the non-fancy engineering version of it. And then this is the sort of the benchtop hollow fiber system. This also has multiple sensors with it and an automated sampling tool. We can also go into the bioreactor, sample media at any time, at any interval, and put it to any instrument that one may want. So very, very sort of one set of data. We have a lot more. But as you would see that for some donors, certainly the bioreactors help over T flasks in terms of growth, but in terms of yield, it didn't matter for one donor, but for another donor, we got a lot more donors, a lot more cells when we had this feedback-controlled closed-loop bioreactor. And the quality does not change, or at least I wouldn't say the quality does not change. I should simply say the surface marker, the identity did not change between the different batches. We think there are other quality metrics that we need to look at, which is what we are looking at. But this is now a fully automated, closed system that we don't need to put inside a GMP room, but it's an end-to-end box that can sense, decide, and control. And we have done that for T cells as well. We have a lot of sensing projects as engineers. This include a lot of flexible electronic sensor that goes into the bag walls. And if anyone's interested, happy to discuss about that. These are multiplex sensors that at the same time does pH, DO, and many other things, flow rate measurements, but sticks to the wall of the bioreactor as a flexible electronic sensors and can wirelessly monitor. Sensors for rapid microbial detection, for inline impedance sensors for cell growth, multiple microfluidic technologies for potency assays, disease-on-a-chip models, and so on. I'll give you two quick examples. This is how kind of the flexible sensors look like. Each of these is a different sensor, and this is fully foldable, you can crumple this thing, and then it still works, right? And so this can go into bags or bioreactor walls. And then this is a project that actually started with Joanne calling me one day and asking that I have all the card bags. I don't want you to open them, but I need you to tell them how much WBCs I have in there, mononuclear cells I have in there. I'm like, okay, so I can't open the bag, and I have to tell you how many mononuclear cells we have. So we came up with that. That is a solution we have now. We need a transparent wall in those bags to do that. But Paco Robles, who is a faculty at Georgia Tech, came up with this oblique back illumination microscopy to quantify very, very accurately the mononuclear cell count in the middle of RBCs and everything else. And this is sort of the resolution they can get, and now they are working on even figuring out metabolic signatures out of this using this oblique back illumination microscopy techniques. And we are working on integrating that with bioreactors as we make them. So overall, the grand vision for us, at least in this Cell Manufacturing Technology Center, is really this process-controlled, full automated systems for scalable manufacturing, where we have enough knowledge and modeling of the process to be able to predict where the cell culture is going and control it, sense it in real time. And on top of that, the other area that we are working on that I didn't have time to talk is really supply chain modeling. This is a huge area that you all know. COVID has told us how critical that is. But even there are other ethical issues that come up. As we go into 100,000 patients, but we have capacity to manufacture 5,000 patients, who do we manufacture first? The person who came in the line first, or the person who's sickest goes first? Who decides who is the sickest patient? So these are questions that companies are starting to deal with now, even for multiple myeloma and others. How to queue patients, in the context of my manufacturing capacity, how much redundancy of suppliers do I need? So if supplier one goes down today, how many patients will die? So those are models that very, very apt at this point. And every other industry has done those models. If a supplier goes down in China, Apple does not shut down their production of iPhones every time, right? So those type of production models needs to be done. And this is, a whole another story that we can chat about later. So I'll stop here. I think we probably want to keep questions till the end, but happy to take one or two.
We have time.
Okay.
A quick question. If anyone has any quick question. I have one question.
Sure.
So you do metabolomics and secretome analysis, and you did talk about how some of these findings indicate if your process is going well or not. Mm-hmm. What's your outcome that you're measuring?
Yeah.
Is that proliferation or just something else?
It's all different ones. So for T cells, we measured proliferation, cell killing, CD4 to CD8 ratio, memory T cells, which was our the data I showed you for TNF and IL-2 receptors are for stem-like memory T cells in CD4, not stem-like, central memory T cells in CD4 versus CD8, because those are correlated to patient function now. For MSCs, we do macrophage polarization, TNF suppression, IDO, T cells, and for each of them, we try to find what is the most consensus signature that tells us that the cells are most functional at the end of manufacture.
Okay, thank you.
Okay. Sure.
That brings us to the last speaker, and Dr. Anthony Filiano will be talking about preclinical studies of cord blood monocytes and monocyte-derived products.
Yeah, thank you. So I switched the title up a little bit. I wanted to really emphasize this fact that monocytes and their related macrophage are a real underappreciated and exciting immune cell that we can use in cell therapy. So if MSCs and CAR T are Facebook, I think monocytes and macrophages are the TikTok of the cell therapy world. So what is a monocyte? I think probably most of us in the room know that monocytes are a very dynamic immune cell. They represent anywhere from 10 to 20% of your total white blood cells, and they're very dynamic. They come in different flavors, and they have a lot of different activities. So I think most of us know how important monocytes are for controlling blood infections, but they can also promote tissue regeneration, and then the related macrophages are present in all tissues. And macrophages are really important for tissue homeostasis. So we want to utilize some of these factors into our cell therapy products, and we really have three different strategies when we think about monocytes. So monocytes, they can release factors, and so that's one strategy we'd like to look at. Another one is that monocytes have the capacity to infiltrate into tissues and differentiate into macrophage. So you can see how a monocyte can be used as a cell therapy product for engrafting to replace macrophage. And this becomes really important and challenging in the brain where microglia maintain their own local niches. So we're looking at different strategies to promote engraftment of monocytes into the brain. I'm not going to have time to talk about that today, but I'm happy to discuss afterwards. And then lastly, another strategy that we have is using engineering to engineer novel monocyte-derived therapies. So I'll talk about the first two in the talk today. So we all know that hypoxia is a pathologic characteristic of obviously stroke and traumatic brain injury, but it's also associated with diseases like cerebral palsy. And so to model this in a dish, we developed a brain slice model where we can deprive it of oxygen and glucose. And this is what the slices look like. You can see these beautiful images with, So these beautiful images of MAP2, which labels neurons, and MBP, which is a component of myelin. And so myelin is the insulation around nerves, and it's incredibly important for nerves to transmit for synaptic transmission, to maintain synaptic transmission at the proper speeds. And then you can see the cell bodies in blue. So when we treat these with oxygen and glucose deprivation, we can see cell death as incorporation of PI. So you can see all of this red staining here. And we noticed if we incubated these after oxygen and glucose deprivation with increasing doses of mononuclear cells from cord blood, we were able to suppress this cell death. Further, what was really interesting is that the mononuclear cells did not need to touch the tissue slices. If we put them in a transwell, they were still able to prevent cell death in this model. So I think from the title of the talk, you can guess what the active component is, but it was really important for us to figure out what type of cell is promoting this, is protecting cell death here. So we did an experiment where we depleted different immune cells in cord blood, T cells, monocytes, B cells, hematopoietic stem cells, and we found that only when we depleted the mononuclear cells of monocytes, CD14 positive monocytes, it blocked this protection. And then conversely, we could isolate components from the cord blood, T cells, monocytes, B cells, stem cells, and then add them to the assay, and we saw that monocytes offered the best protection. There was also a little protection from CD34 positive stem cells, but there is a component of monocyte progenitor cells in that pool. So if we used adult peripheral blood mononuclear cells, we found that they failed to protect. So you could see that here's our protection with cord blood, MNCs, where the adult failed. And even if we isolated monocytes from adult blood, it also failed to protect. So what's the difference between cord blood monocytes and adult? We did some very classical sequencing assays and looked at the differential expression of genes. And you can see just each row of this is, so each column is a different sample and each row is a different gene. And what they are is not important, but you can see that there's major differences between cord blood and adult blood monocytes. And I pulled out a few that are interesting to us. IL-10 is very anti-inflammatory. MMP9 is needed for synaptogenesis, as well as TSP1. And these are a few candidates that we're starting to explore. So cord blood monocytes, they're distinct from adult blood monocytes. They protect in our model of oxygen and glucose deprivation. And cord blood monocytes, they offer this protection by secreting some factor. And we'll explore this later on in the next part of this talk here. So looking at engineering cell therapy products from cord blood monocytes. So in 2015, the Marcus Center developed what's called a DUOC-01. And so this is a macrophage-like cell that's derived from cord blood monocytes. And so you can see from this photomicrograph that it's a very heterogeneous group of cells. So if I showed you flow from these, it expresses all these markers that macrophages have. But you can see there's these very string-like cells that look more like a cultured microglia, and then there's these other cell types that look more like phagocytic macrophages. So we wanted to dive a little bit deeper than this just visualization of the bright field image. And we did single cell sequencing. And using this resolution, we were able to pull out multiple different clusters. So we just take this culture, run single cell sequencing, and look at this clustering of the culture. And we were able to find that these certain clusters, a lot of them were maintenance, basically like maintenance clusters that were enriched for pathways like cell division and DNA synthesis and repair, ATP metabolic processes. And then there was some other clusters that were enriched for pathways that you would expect, like antigen presentation and a lot of these immune-based pathways. One of these clusters that we were really interested in is this one up top, and you can see that I just wanted to highlight some of the enrichment pathways that popped up were lipid processing and myelin. And so for someone who's interested in CNS autoimmunity and diseases of myelination, this was really interesting to us. So in addition to this single cell sequencing to characterize the cultures, we had developed a signature that we could use to detect microglia and then also monocyte-derived macrophages that engraft the brain. And so I don't have time to really go into this, but we developed these signatures based on multiple different experimental models and then published available data sets. And you can see by immunohistochemistry that, IBA1 is just a general macrophage marker. And you can see the GFP in this experimental system is hematopoietic-derived macrophages. And they look very similar, but we could use this signature that we developed to be able to identify the two. And we did this for an experimental system, but we applied these signatures to these DUOC01 culture, and this is just a test data set showing how the signatures were not present in these other immune cells, but in microglia, they were enriched in this microglia signature set. When we applied it to the DUOC01 culture, we saw that they were enriched for both signatures, both the microglia signature and this brain engrafting macrophage signature. So this suggested to us they may be useful for looking at diseases of the CNS. So we're very interested in the CNS autoimmune disease multiple sclerosis. Many of you are probably aware of the symptoms and the etiology, but this is a mouse model called experimental autoimmune encephalomyelitis, or EAE. And for this model, we inject mice with myelin oligodendrocyte lipoprotein, which is a peptide component of MOG. And so this is the antigen used in this model. When we immunize the mice with adjuvants, the mice end up developing autoreactive T cells that attack the myelin sheets. And over time, they end up developing this ascending paralysis that starts in the tail and works its way up. So in this assay, we immunize mice, and then we wait for about day 10 to 13 when the mice develop paralysis. And then we inject our cell therapies or controls into the cerebral spinal fluid at the third ventricle at an area called cisterna magna. You can see in the mouse here. So it's a really nice, a pretty non-invasive spot for injecting into the CSF. So these are what the data look like after the mice hit a clinical score of about 0.5 to 1, which is minor tail paralysis. We come in with our groups. We injected ringers and DUOC01 into the CSF. And then we also included hydrocortisone in these studies in case there was some clinical adverse events where we needed an anti-inflammatory. So we unbiasedly selected mice into these 4 groups, all at the same scores. And you can see when we injected DUOC into the CSF, it prevented the progression of this clinical score. And hydrocortisone did not have an effect in this model. So this is a very nice PI experiment where you could, I talked to Lee, a talented research scientist in the lab, and I said, let's do this experiment. You can go and you can watch the mice every single day, and we can just closely monitor them until they hit that score. And then I go back in the office and do PI stuff, and she's stuck looking at these mice every single day. So a patient with MS, they're usually not diagnosed until their symptoms have gone on for a few months or a few years. So it really wasn't realistic for a therapeutic. It was more of a preventative strategy. So we shifted gears a little bit, and we waited until the mice hit peak of disease, which is about day 27 or so. And then we injected DOC-01 as a therapeutic dose. And we were really excited here, and because the mice all had, they had similar clinical scores, but it was ranging, we normalized them all to, and then the data I'm presenting is a change in that clinical score. And we were excited to see that when we injected DOC-01 in these mice, it was able to decrease ongoing clinical paralysis. So again, the scientist in me, what's the mechanism here? So we went back to the lab and started looking at this. And for EAE, it's a CNS inflammatory disease. It's mediated mostly by CD4 autoreactive T cells, but there's huge inflammation in the meninges and infiltrating cells into the spinal cord. And so if I had to put money down, I would have guessed that DOC-01 were inhibiting these pro-inflammatory cells from coming into the CNS. It didn't, as you can see here. And there could be a couple reasons that we're still exploring. But other potential mechanisms, they had this robust effect of protection on clinical paralysis. So one, it could be that they're altering the skewing. It's not preventing cells from getting there, but it's altering the types of cells that get there. Another thing is it could be limiting their activation. And then one of our favorites is that it could be promoting recovery and promoting remyelination of the CNS. So we shifted into another model that's better at looking at remyelination. So this is a cuprizone-mediated model of demyelination. So you feed a mouse cuprizone. There's still a little ambiguity of what cuprizone does, but we know that it is detrimental to oligodendrocytes. And you can see this very profound demyelination in the corpus callosum here. So these are brain slices, coronal brain slices, stained for MBP, myelin basic protein. And so you can see with cuprizone, you have this very severe demyelination. When you remove the food, you get spontaneous remyelination, but it takes a long time. So again, this talented scientist in the lab, Arjun Saha, injected DOC-01 into the corpus callosum of these mice. So this is ringers alone. When he injected DOC-01, you could see this very robust increase in remyelination in the corpus callosum. So again, getting more at the mechanism, we needed another assay that we can manipulate a little easier than the mice. So Lee developed a slice culture model system. So you can see these little brains floating around, little slices of brain. And you can see the beautiful architecture over time. So you can see the cerebellum here with the gyrus. And all these beautiful CNS cell types are in here. So red are neurons, purple, magenta are astrocytes, and you can see green microglia. And Lee stained these sections with neurofilament heavy chain in the red here, and you could see all these beautiful axons. And when she co-labeled with MBP, you can see they're nicely wrapped with this myelin, and in the merged image, all the ones in yellow. We can use a different reagent to demyelinate these. This is LPC. So this is control. We can use LPC here, lysophosphatidylcholine, and you can see it just massively demyelinates these slices. And then when she put DOC-01 onto the slices, when she washed out LPC, you could see this very nice remyelination of these axons. And when she quantified that, you could see here, so this is the amount of MBP that's co-localizing with the neurofilament. So increased number here would be more co-localization or more myelination. So with LPC, as you would expect, you can see this robust demyelination in these axons, and then with DOC-01, it really promotes this remyelination. So again, we really like assays in lab. And each of them, I don't have time to talk about the advantages and disadvantages to each of them, but this is a very nice assay that we're looking at developing into a potency assay because Lee has really worked out the assay to its very simplest form here now. But for this one, we wanted to see if these cord blood macrophages could have any direct effects on oligodendrocytes. And so oligodendrocytes, you can see kind of in this cartoon here, is mature oligodendrocytes derived from oligodendrocyte precursor cells in the brain. So it's these rapidly dividing cells, especially when there's injury. And they go through this maturation process to mature OPCs that end up making myelin and wrapping around the axons. And what's nice is we have these different markers that we can see what stage these OPCs are in their maturation process. So for this assay, she isolates these oligospheres that are pretty easy to maintain in culture. She can dissociate them into single OPCs and put them in a differentiation media, and they'll mature into myelinating oligodendrocytes over about a week or so. And then we can look at their progression with these markers. So when she took these OPCs and differentiated them and added DOCO1, she noticed that a lot of these cells end up expressing these mature oligodendrocyte markers like GAL-C, and that's quantified here. She also saw these really beautiful oligodendrocytes that expressed MOG, which is what's a component of these myelin sheaths. And so what's kind of always been in the back of our mind here, so if these macrophages, when they're injected into the CSF, and they have such protective effects, and then we see in culture that they have all these effects on pushing the maturation of OPCs to mature oligos. How is this happening? Because when you inject cells into the CSF, there's a lot of neuroanatomical borders that separate CSF and interstitial fluid from the brain parenchyma itself. And even if you label these DOCO1 cells and inject them into the CSF, you never find them, it's rare that you would ever find one in the parenchyma itself. And you can see really any cell type, not just these DOC, even in inflammatory conditions. But where you find them is in the meninges surrounding the ventricles, and this is the pia layer of the meninges. And so we have this hypothesis that these cell therapy products are not promoting protection by physical contact on these cells, but it's really changing the CSF milieu by secreting factors and changing the CSF to an environment that would promote the maturation of oligos and remyelination. So we wanted to test this in an assay. We went back to our transwell assays where we can have our macrophages in the transwell and then have our OPCs underneath. And just like I showed you before, when you add DOCO1 to these OPCs, you see these very beautiful, mature oligodendrocytes start to form. And that's, again, quantified here as the amount of cells that express this MBP. When we used a transwell assay, we saw that we could see similar amounts of these mature oligodendrocytes in the assay, suggesting to us that the DOCO1 did not need to directly contact the OPCs, but they could ensure their maturation by secreting some kind of beneficial factors. So this is very exciting to us, and we're kind of moving on to a second-generation product that we call RemiMax. And so this stands for remyelinating factors secreted by macrophages derived from cord blood. And so for this assay, we took a DOCO1 and we isolated the RemiMax from the culture. And then we could use RemiMax as a novel second-generation cell therapy product and tested them in an OPC maturation assay. And what we found, like before, the DOC1, again, promotes maturation of OPCs into mature oligos. But we were really excited about the fact that these RemiMax not only improved the maturation of oligos, but it increased their maturation by about 2.5-fold compared to regular DOC themselves. So the take-home from this is DOCO1 is a cord blood-derived macrophage that's beneficial in multiple preclinical models of demyelination. And they offer this protection by secreting these factors we're calling RemiMax that we're developing into a second-gen product. And I just want to, kind of what we're thinking about the advantages of these RemiMax is, one, we can manufacture them at large scales. If you think about macrophages, especially in culture, there's very little proliferation. So you're kind of limited to the numbers. You're limited really to your input as far as your output. With these Remimax, macrophages are constantly releasing these products. And so we have the capacity to produce a large scale. Two, they're a cell-free product. So even though proteins and small molecules can induce an immune response, we don't have to worry about some of these unmatching problems that we have with cell therapies, allogeneic cell therapies. And one thing I like about these Remimax is they're really amenable to continuous infusion. And because we can manufacture large scale and we're infusing into the CSF, we're looking at exploring different techniques with microfluidics to offer continuous infusion of these. So with that, the most important slide of my talk is the acknowledgement slide, and I'd like to thank really all the members in the lab. Most of this work was done by Li Shu, who is here. She has a poster tomorrow, I think, so go talk to her about that. Another postdoc, Hyungjung Min, that I'd like to mention, she also has a poster on some exciting data that we have using mesenchymal stromal cells and this very interesting mechanism involved around a special organelle called a processing body that we're thinking about using for cell therapy. So you want to talk to her about that. The alumni, collaborators, I'd like to thank a lot of the folks in MC3 who have contributed in different capacities on these experiments. Our funding, and I just wanted to definitely acknowledge the Cord Blood Association Foundation, who graciously funded our lab, and that has really been critical for my development as an early stage investigator. So with that, I'll take any questions. Thank you, and I look forward to more discussion.
Thank you, Tony. Any questions? I have one question. So you talked about how you can actually produce these soluble factors and have the same effect, actually maybe even better effect. But you mentioned that you're going to give that through into the CSF. Mm-hmm. Why can't you just infuse it through the blood?
Yeah, there's no reason that we couldn't. We have not tried. We have been using, my lab is interested generally on meningeal immunity and CSF components. So we've been thinking about them as a way to control, basically rejuvenate toxic CSF. So that was kind of where our initial experiments begun. But there's a recent study, I don't know if with Tony Weiscore's work, looking at young CSF into old mice and how infusing young CSF into old mice can promote remyelination and aging. And so we kind of model some of those studies on those data, but it's a good suggestion and we should try it.
Yeah, because I wonder if the soluble factors that you talk about can cross the blood-brain barrier, and this way you don't have to do...
Yeah. Volume, is an issue too. Not an issue, but it's a variable. So, a mouse has 36 microliters of CSF,
Correct.
whereas it has, a mil of blood. So.
Correct. The other question was, how do you explain why the soluble factors with the Remimax, you've seen better effect in myelination compared to infusing the cells themselves? What's the explanation?
Yeah. So, we don't know, but my guess would be is probably the biodistribution. So when you inject any cell really into the CSF, it's rapidly cleared. And we understand a little bit better about CSS dynamics and drainage through different systems like lymphatic vessels that are in the dural meninges of the mouse and in human. So, my guess is when you inject these cells, they either die over, hours to 24 hours, or they're drained through the lymphatics and they're gone. And we've been seeing, especially with the MSCs, we've been seeing effects where they can reprogram immune cells and have long-term effects even after they're gone. But we think what's happening here is that they're secreting actively secreting these factors. And the factors may stick around for a while, but eventually they're drained out. And so that's why we're always concerned about manufacturing and dosing and having a therapy that gives us the option to increase dosing as our top priority.
Thank you. Okay,
Thanks.
I think we have 9 minutes. Any questions for the panel here? And if not, I think we can end earlier. Thank you.